Hello, everyone, and welcome to today's webinar, Impact of Global Surveillance on Diagnostic Tools. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Global Scientific Affairs Molecular Diagnostics at Abbott. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This presentation is educational and thus offers continuing education credits. Please follow the process to obtain your credits by clicking on the Continuing Education Credits window. I'd like to now welcome our speaker, Dr. Gavin Cloherty, Head of Infectious Disease Research, Diagnostics at Abbott. Dr. Cloherty, you may now begin your presentation.
Thanks for the kind introduction, and thanks everybody for joining us today. The title of my talk is The Impact of Global Surveillance on Diagnostics. The agenda, what I hope to cover today, is to discuss our global surveillance program that looks at HIV, hepatitis B, and SARS-CoV-2 genetic diversity, some molecular epidemiology that we've conducted, and then give a brief introduction to Abbott's new Pandemic Defense Coalition that just launched recently. So Abbott has been conducting global surveillance on HIV and hepatitis for 28 years, for over 25 years. More recently, we've introduced SARS-CoV-2 into surveillance. I think for obvious reasons, there's been a lot of attention in the media looking at variants, and so we're keenly interested in understanding how these variants might impact our diagnostic tests. So the premise of this program that's been running for almost 3 decades is to monitor the emergence of new variants, to establish well-characterized specimen types and specimen banks so that we can use this information to make very robust diagnostic tests so that we understand what strains are circulating, make sure that the tests that we have on market work no matter where an infection is acquired, no matter where the test is performed. And we also share this information with the field. We've published more than 125 peer-reviewed papers, and some of them more recent ones we'll review later in this talk. We have a specimen bank of over 70,000 rare specimens that have been collected from 45 countries on 6 continents, and that number continues to grow daily. So looking at some of the milestones of this program, it was initiated in 1994, primarily focusing on HIV. And actually the genesis of this program was HIV Group O, which proved problematic for first-generation diagnostic tests. This was, in a way, a global wake-up call, which Abbott took very seriously and implemented this program to make sure that we would be aware of how HIV was changing over time and in different geographies. And as you can see in this timeline, in 2009, we added HBV to the global surveillance program. In 2011, we identified the case of HIV Group P, which we were only the second group in the world to ever find this group, this novel variant group of HIV. In 2014, we added hepatitis C, which was again another important milestone in the program, which also coincided with a lot of new direct-acting antiviral drugs coming on the market. Very important that all the diagnostic tests function as they're supposed to around the world. In 2015, we actually identified a novel human virus called human Pegyvirus 2, and we found that in patients who had hepatitis C predominantly. It was an interesting observation. We also started looking at hepatitis delta as a co-infection for hepatitis B, and pegivirus was also added to our surveillance program. So now we had really hepatitis B, hepatitis C, hepatitis delta, and this novel pegivirus also added to our surveillance program. We took a more public health approach as well in making use of the work that we were doing around the world to understand the molecular diversity of these viruses, to also help inform on prevalence and some public health aspects of these epidemics regionally and in specific countries. As I mentioned, we've also in 2020 added SARS-CoV-2 to the surveillance program. So why is a global surveillance program important? And HIV is a nice illustration. Number one, it's the virus that we've been following and studying, studying the most carefully. But this graphic kind of illustrates it very nicely, that there's been 4 distinct zoonotic transmissions of HIV through human history, and illustrated here with group M, N, O, and P. And if you take group O and group M subtype B, they're really very, very different viruses. And at the amino acid level, they're only 50% similar. So really having tests that are able to accurately detect and quantify such diverse viruses is extremely important. And this program has enabled us to understand that level of diversity in a lot of circulating recombinant forms that are now more predominant in various parts of the world, and ensuring that diagnostic tests work all the time. In addition, we're showing here 4 separate zoonotic transmissions. You have 45, non-human primate species that could potentially be reservoirs for other zoonotic transmissions. So we've got to be ever vigilant in our work. The impact of not being vigilant is quite stark and quite sobering. So this slide starts to show a case study of a group P that was identified in France, which really, was very surprising. In 2004, a 62-year-old Cameroonian woman who was residing in France came and was diagnosed as being HIV positive in a Paris hospital by an antibody test, and it was Western blot confirmed. But when the physician went to conduct a viral load test with the monitor assay that was available at the time, there was no detectable viremia in the blood. So this was puzzling, and there was several draws and tests performed over time. So the lab had also the Abbott RealTime HIV assay available. So they then tested these samples with the real-time assay, and they found a viremia of about 4.4 to 5.3 log copies per mil. So this woman did have an active HIV infection and confirmed the architect combo result as positive. The lab tried to do some additional investigation to understand what was going on, to try and see if it was maybe a group O infection or some divergent group M, and that may have some mutations to the primers and probes. And what actually they found was quite surprising. They had successfully amplified and sequenced the genome of a new group, a group P, which came from gorillas. And that explains the image here on the left-hand side, where you've got the Eiffel Tower indicating Paris and the gorilla indicating where this group P had originally come from. So what's impressive here, in addition to the discovery of a new HIV group, is that the architect combo assay and the Abbott RealTime viral load assay both worked in this very divergent virus. Without any pre-existing knowledge of that virus or its sequence, the way these tests are developed, relying heavily on our experience with this global surveillance program and knowledge of the diversity of these viruses, enabled us to build a test that was resilient and robust enough to pick up this new group of HIV. So kind of a testament to why we do this surveillance program. In addition, there was some media interest in this paper that I'm presenting here, where through work we had performed in the Democratic Republic of Congo, where we were sequencing HIV and hepatitis strains found there, and we had processed about 10,000 samples, we actually identified HIV group M subtype L, which enabled this subtype to be officially classified. And this was the first new HIV subtype that had been named in 20 years. So this was a kind of an important event and created a considerable amount of media interest. Also important to note that, again, all the Abbott tests were able to detect this subtype L. So it's not, as I mentioned, just HIV that this program focuses on. We also look at hepatitis B, hepatitis C, SARS, and Hep Delta. I'm highlighting here an interesting paper where we looked in samples we had received from Thailand, and we were able to identify a novel genotype I, which is a very rare strain, and it had not been previously characterized in Thailand. So this now is present in Thailand and may be circulating, and would instigate additional interest and research into understanding more of the genetic diversity in this part of Southeast Asia. So for hepatitis B, just like HIV, there is a danger in not being vigilant, in not developing tests that are to genetic diversity, not understanding that genetic diversity that may be out there. And so in this study that was published, this is not an Abbott study, but it was published by Hendrickson and al. They looked at hepatitis B in dialysis patients, and they found a case of a patient who had been vaccinated effectively, had an anti-HBs titer of 82, so should have been effectively vaccinated, and was hepatitis B surface antigen negative when originally tested. And so this person then would have been dialyzed on common units, instruments with other patients. And later on, it was identified that this patient was surface antigen positive when they were tested with the Abbott Architect platform. And when they went back, they found through archive samples multiple instances where this person had been positive and had not been detected using a different test. And the survey found that 9 out of 23 institutions or labs that were testing these types of samples did not use tests that were robust to common surface antigen mutations. So again, I think it's important to be ever vigilant, because if you're not, you could, in this case, risk transmitting the virus to other people who are at risk, or missing infections and missing opportunities to treat people appropriately. Moving on to SARS-CoV-2, I think anybody who's not familiar with SARS-CoV-2, or where it came from, must be living in a cave for the last year. But this slide does illustrate that it's a novel beta coronavirus. Incubation period is roughly 5 days, but anywhere between 2 to 14 days. Not always symptomatic, but symptoms usually happen around 12 days, so 10 to 12 days. And so you can have mild to moderate disease or hospitalization and fatalities, so deaths, 2.4 million people worldwide. It's a real, as everybody's aware, this is a tragic situation. So one of the earliest efforts and ways to try and tackle the pandemic that was brewing was to develop an assay, a molecular assay. And in this case, we're describing here the target selection of the Abbott molecular assay. And we selected a dual target both for robustness and redundancy. So in case there were mutations in any region that might impact one of the regions, the other would be able to amplify. We selected nucleocapsid and OD/P region of spike. And again, keep in mind that we developed this assay with one sequence. So we had just that initial sequence that was released. We used that and the knowledge that we had accumulated over the 30 years of studying diverse viruses and making diagnostic tests to select these two regions that were the most conserved, and we figured would be the most conserved moving forward, and that's proving to be the case. So there, as I mentioned, there's been a lot of interest in SARS-CoV-2 variants that have made a lot of impression on the media. We've heard about the UK variant, the South African variant, the Brazil variant, and these we have evaluated very carefully in the global surveillance, in our global surveillance program. The mutations are illustrated here on the right-hand side. They range from nucleocapsid, spike, open reading frame regions. Some are in E as well. But what's most important from our perspective is that we've analyzed in silico through bioinformatics pipelines and predictions, et cetera, that the Abbott tests are not impacted by these mutations. And actually, in the case of the UK variant and the South African variant, we've physically tested samples containing these variants and found that they're accurately detected, that there's no impact on the performance of the test. In addition, we're continuing to be ever vigilant and sequence samples that we source from all over the US and globally, and some are illustrated here in South Africa. We're collaborating with CRISP in South Africa, working with folks in the United Kingdom, in Senegal, in Brazil, in Canada, to really keep an eye on how these viruses are changing over time. And that list continues to extend as well. This is just a representation. In Illinois, we've been sequencing, actually found some UK strain floating around it in the UK, in the Illinois population, I'm sorry. Sequencing in California through our collaboration with UCSF, sourcing samples from Wisconsin and Michigan, and putting that with Chicago to get a feel of what's going on in the Midwest, and sequencing samples from New York, where we found the New York variant, as well as the New York variant with the E484K variant as well, which is, again, in quite a few samples, so it's a concern. So this is an ongoing effort. And on the right-hand side, I think we'll see a graphic of the clades and diversity we found in our early study, basically back in March, to see what was circulating. And what was interesting is that the clades, the samples that we sequenced from the East Coast and Michigan reflected more of the European kind of feel, and the ones we sequenced from further west were more of the Asian feel, and then Chicago was a bit of a melting pot of both. So it was an interesting illustration of human movement, I guess. And we've developed novel techniques to make sure that we're able to sequence even low titer samples. So we've applied what we call an X-gen approach to HIV and hepatitis, and that's using an enrichment, using probes, oligos that are specific to that pathogen, to kind of pull them out preferentially and sequence them. And we do that in samples where we've got a low titer, and it enables us to get whole genomes in about 90% of cases, or even more, down to about log 3.5, and most of the genome in samples that are even lower viral loads than that. So how does the program works? Through partnering with collaborators around the world. And some of them are listed here, and you can see it's very diverse from South America, Asia, Africa, Europe. It's important to be aware that we can't do this on our own, and we need to partner with collaborators all over the world to understand how viruses are changing in real time and across the globe. And the pathway we use, as I mentioned, was collaboration with people who have access to patient samples, whether they be hospitals or blood banks. We put these samples through a very rigorous testing algorithm, whether it be with our diagnostic tests, in-depth sequencing, other analysis should we find something interesting. All is part of the package and the process that we put in place. And then if we discover something, we make that available, that information available to our collaborators and also to the general space through peer-reviewed publications. As I mentioned, we've got 25 now, which I think might be an underestimate at this point. But so we do share our data with our collaborators, or the data with our collaborators and the space. And then we use this information, it's not just an academic exercise, to constantly refine, improve, design either our existing tests or new tests that may prove useful. So it's important to keep in mind that these viruses are constantly evolving, so our diagnostics needs to constantly evolve to keep up. And switching gear a little bit to molecular epidemiology, I think it's important, as I mentioned in a previous slide, to keep in mind that we can do more with the information that and the samples that we're collecting and the work that we're doing around the world. So one effort is to try and dovetail this to get some epidemiology information out by partnering and collaborating with other investigators. So I mentioned previously, we talked about the HIV group M subtype L that we found in the Democratic Republic of Congo. And through our collaborations with Carol MacArthur at UMKC and the University Protestant of Congo, we've been able to characterize and test more than 10,000 samples that have been collected over a few years. And one of the things that we observed through our diagnostic algorithm was that there was a discordance between the number of HIV combo positives and HIV viral load positives. So we had a higher proportion than expected that were antibody positive, that we couldn't get any viral load or was very low. So we started to investigate to make sure that there were no issues with our diagnostic tests, doing Western blots and different PCR tests and using comparator assays and stuff. And we found what was quite interesting. We found a very high percentage of these samples looked like they came from elite controllers or suppressors, so people who were able to naturally control the virus themselves. Because as part of the enrollment, there was a questionnaire that said, were you on therapy? But just to be on the safe side, we also sent samples for mass spec to see if there were any evidence of antiretroviral drugs on board. And we did find that actually quite a considerable percent, I think 40 to 60% of them, did have evidence of antiretroviral drugs, so they were excluded from the analysis. But even still, we found approximately 2.7 to 4.3% of these samples who had no evidence of antiretroviral drugs, but were either low or negative for viremia and truly antibody positive. So these would fit the criteria of presumed elite suppressors. Now, what we need to do to explore this further would be to get longitudinal samples from these cases and show that they are continuously suppressed, and also, would afford us the opportunity to look at the immunology of these participants to see how exactly are they doing this. Normally, you might find 0.1% of a given population might be able to control the virus naturally, or in some cases we've seen in the literature up to maybe 1%, but never up to 4 or 4.3%. So it's a quite phenomenal finding that we were able to, observation that we were able to make through this study. In addition, we've been collecting samples for HIV surveillance in Cameroon for many years. So we availed of this to go back and look and see how much hepatitis C we could find, or how much of this new PEGI virus we were able to identify. And so what we found was quite interesting, that the percent of hepatitis C was roughly about 2.5%. And I had heard, anecdotally that it would be anywhere from 11 to 15%. So this was quite interesting to show that with an RNA test, so not using any antibody tests or lateral flow rapid tests, we were able to show that the prevalence was about 2.5%, and this was year over year. So these studies were done over many years, and the data was consistent. And the samples were collected from a geographically diverse sites around South Cameroon. So what we also found was that there was a predominance of male. So you'd have up to 3.8% positivity in men versus women were about 2%. And also that there was an age bias. So while most of the samples were coming from younger people, let's say in their 30s, maybe down in their 40s, most of the positives were coming from people who were in their 60s, 50s, 60s, and above. So there was definitely an age cohort effect here, and also gender bias. So it was very interesting data. And we also found evidence of the novel virus among people who are HIV, hepatitis B, and HCV positive in Cameroon. But again, most of the positives came in people who were HIV and Hep C positive. We also then decided to leverage again these samples that we had collected from Cameroon and look at about 1,900 samples that had been collected that were hepatitis B surface antigen positive, and see if they also carried a hepatitis delta, evidence of a hepatitis delta infection. And we found, to our amazement, that 46% were antibody positive for delta, which led us to then interrogate further to see in these samples how many were hepatitis delta RNA positive. And we found that almost 35% of the Hep Bs were Hep delta positive. Now, this is incredibly important from a public health perspective, because the prognosis for a Hep delta HBV co-infection is quite poor, with many or most going on to develop cirrhosis and potentially cancer, and high rates of morbidity and mortality. So it's important to be able to make the Cameroonian folks aware of what we had found, and so we published this data. It was other data was later reproduced by, I think, Institut Pasteur, looking at the entire country of Cameroon, and they reproduced what we found in South Cameroon, and actually found that North Cameroon, the prevalence of Delta was actually quite a lot lower. So it's important to look to fine-tune the geography that you're searching. We've also been busy looking at novel biomarkers for hepatitis B, trying to facilitate a greater understanding of this virus, and maybe enable novel curative therapies or different approaches to treating that virus. And in this slide here, we see, the life cycle of a hepatitis B virus as it enters the cell, moves to the nucleus, gets transformed, unpackaged into covalently closed circular DNA, and then is transcribed and translated. It also shows where the standard of care nucleoside analog therapy interacts and interrupts the life cycle. So it really impacts at the end, where the hepatitis B RNA is converted into DNA before being released into the blood as a gain particle. So what's important here to keep in mind is that this therapy does not impact anything that happens upstream. So it has no impact on the generation of hepatitis B RNA, its encapsulation, and potentially its release into the plasma. So we developed a test to have a look at that. And on the right-hand side here, you'll see with a standard HBV DNA assay that the usual trajectory, that the viral load increases to a set point. So you've got a DNA viral load level, and then as you apply a nucleoside analog therapy, the DNA declines. But as I mentioned previously, this nucleoside analog therapy has no impact on the production or release of HBV RNA. So RNA actually can remain pretty stable over time, and then might decline as you get an immunological response or some other mode of action that can impact the course of the infection. Okay, so I think here I want to show the assay that was developed for the M2000 assay. It's a research use only assay, and it's designed to be automated. We standardized it to the WHO DNA standard so that we can have some kind of understanding of the units and what we're seeing. So 1 unit of RNA is roughly equivalent to 1 IU of DNA based on the WHO standard. The limit of detection of the test is 44 units per mil, and we published these findings in Hepatology in 2018. Important to note that we actually have a dual target approach here too, and that we did this for multiple reasons. One was so that there would be redundancy in case there was any mutations that would impact the detection and amplification of this target. The other was to try and understand if what we were finding in the plasma was entire pregenomic RNA, or if it was pregenomic RNA and other subspecies of RNA. The expectation there is that if there were these other species present, that we would be able to see it by a divergence in the signals that we got from the X and the core, because both of them have different fluorophores. By comparison, this automated test is significantly more sensitive than the previously manual race-based method that was published by Van Bommel in Hepatology, had a limit of detection of roughly 300 copies per mil. And so there's been a lot of research done to understand the relative performance of these two tests and the clinical utility, and I'll talk about that just shortly. So one interesting question that had been posed earlier was whether RNA was present in the plasma as kind of like an accident, or was it there, you know, routinely and reliably. So we looked at a couple of seroconversion panels, so where we had sequential samples from patients who had become infected as they went through the course of either resolving that, as in the top two panels here, or going chronic, as in the bottom panel. And what was remarkable for us to see was that the RNA was present almost from the start. And it was present at about 1.5 to 1.7 logs lower concentration than the DNA. So it was there in not as abundantly, but it was there from the start, which would imply that this is a routinely found in plasma and was not some kind of accident, a biological accident. It's also interesting to see that, you know, when the patient goes chronic, that the RNA tracks very closely with the DNA in terms of how much is found in that patient. And also that the X and core targets were found in pretty much the exact same concentration. So it did not appear that there was any difference between pregenomic RNA or pregenomic plus, which implies that the predominant species of HBV RNA in the samples is pregenomic RNA. So looking at the clinical utility of this marker, a study that was published by Carey et al. From King's College looked at patients who were on nucleoside analog therapy, were suppressed, and whether the question from the physicians is it safe to stop nucleoside analog therapy? Because there can be the risk that the liver will flare, that you'll get a flare which can in fact be quite serious and even fatal. So they characterized a flare as no to minimal, as less than 2 of the upper limit of normal. Whether it's mild would be between, you know, less than 5 upper limit of normal, and greater than 10 was a severe flare. As you can see on the left-hand side, hepatitis B DNA was negative by default. So the person was on nucleoside analog therapy and they had been suppressed for some time. So this marker is no longer telling you anything and does not predict whether or not somebody would have a flare. But, and similarly, surface antigen was also positive in all of these samples at a very relatively similar rate. So again, did not predict a flare. However, the HBV RNA assay, when it was positive, was a very good marker of whether or not somebody may experience a severe flare after stopping therapy. So in the patients who had no flare and those who had a mild flare, the RNA was not detectable. But in all cases where there was a flare, RNA was still detectable and quantifiable in these cases. That shows that replication was still ongoing in the hepatocytes, and so these people were at risk of a flare. In addition, a similar study, there were 19 patients who were on long-term suppressive nucleoside analog therapy. They had a negative DNA. They were stopped again. So the question is, could pregenomic RNA or the presence of anti-HBs be predictors of who might relapse? And as you can see here, the hepatitis B DNA was negative in those who, in the preceding samples, in those who relapsed. Also, there was no benefit to looking at surface antigen. I'm sorry, not surface antigen, core antigen. But in this case, where HBV RNA was still positive, both of these patients did relapse. And also what was interesting was that when you had a robust anti-HBs result, they did not relapse. And the patients, the two who relapsed, did not have a very robust anti-HBs response. And they were also viremic here when you see the HBV RNA results. So in short, the HBV RNA seems to be an interesting tool for studying the natural history of hepatitis B. It does appear to have clinical utility in predicting whether it's safe to stop somebody who's on a long-term nucleic analog therapy, and whether or not the person should be stopped in terms of their risk of having a relapse over time. So if RNA is still positive, these people may be at risk of an ALT flare, and they may be at risk of relapse, so it's probably best to keep them on treatment. This test is also being used extensively in clinical trials to try and understand its role and performance with novel therapeutic compounds and approaches. So moving to a slightly different topic, I want to make you aware of a program that Abbott has launched in the last couple of weeks. Now, the name might imply that this is a response to the SARS-CoV-2 pandemic, but in fact, this program has been in development for several years, and in fact was delayed by the pandemic. The launch was delayed by the pandemic, so the time is right, I think, for this to take flight. So it's called the Abbott Pandemic Defense Coalition, and the intent is to, as the name would imply, to try and prevent another pandemic from taking hold. So we've seen the effect of the current pandemic. We have Ebola now, popping up again in the Democratic Republic of Congo. Zika ripped around the planet a couple of years ago. We had SARS, the original SARS and MERS outbreaks. So these are coming at a increasing frequency, and we've had a couple of near misses in terms of pandemic profiles. There's also an amazing amount of undiagnosed illness. So if we look here in acute respiratory cases, maybe 15 to 30% are undiagnosed, up to 70% of meningitis and encephalitis. Gastroenteritis, up to 50% go undiagnosed. So is there a pathogenic or a pathogen that could be the root cause of all of these illnesses that go undiagnosed? These were the questions that were on our mind as we were building the capabilities to launch this coalition over the last several years. So it's important to just make the point that this is not an academic exercise. This is a scientific exercise, sure, but it's really a public health partnership that's dedicated to try to identify early some threats that could pose a risk of a pandemic and to respond to try and prevent that. So outbreaks might happen, but hopefully we can prevent a pandemic. The approach that we're taking is a holistic approach. So as many others are, we're looking to see if we can find pathogens that may provide a fertile ground for a potential outbreak or pandemic. But in addition to just identifying these threats, we're going to be very active in rapidly developing molecular and serological tools through, whether it be through core lab or rapid tests, that can then be deployed to our partners around the world to try and understand what we found. If we identify something in Africa or in Thailand or in Brazil, do we see it in Chicago? Do we see it in France? Where else in the world do we see it in Australia? Where else do we see these evidence of this pathogen? So that kind of translational research is really critical to inform public health to the level of risk. And then if we do identify something that is a potential risk, we can then manufacture these tests on scale as we did for SARS-CoV-2 and try and, prevent an outbreak from becoming an epidemic. And I guess, I was asked before what a potential book, if I was to write a book on this in a couple of years, and I think the title of it would be Pandemics That Never Happens. That would be the title that I would hope we would have. So the network will include infectious disease experts around the world. And the requirement is quite high. We need these experts because we need to identify unique cases that are unusual, cases that are a potential risk, trends among populations, things we might want to investigate further. We also want to partner with labs and collaborators that have a lot of technical expertise. In some parts of the world, samples can't be shipped. We need to be able to do this work on site. And actually it's preferable in terms of timeliness to be able to do this work locally. So that's another key characteristic that we're looking for in partners that we're joining to date. And so in summary, we talked about the need for elite infectious disease expertise and a high level of technical expertise to do the work locally where possible. It's also important that everything is done in a very controlled, systematic, and high quality way, from sample collection to storage to shipping, bioinformatics. So we've invested a lot of time and effort in building robust bioinformatic pipelines that will enable us to identify these potential novel pathogens and also enable us to rapidly prototype tests that can be deployed both for the purposes of understanding the nature of what we've discovered, and also from deployed for public health protection, if you like, so that we can try and ring fence any outbreak and prevent a next pandemic. And speed is of the essence. So I think this is a critical point to make, that we don't have weeks and months and even years, as sometimes is the case when you're developing tests for routine diagnostics. We have to be able to move fast. So I appreciate your time, and I think that's all I have to present today. Thank you.
Thank you, Dr. Cloherty, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Let's get started. Our first question is, you mentioned that you used one SARS sequence for your molecular assay. What are some of the attributes you look for when designing assays with limited sequence availability?
Well, yeah, that's a tough one. You try and draw on the knowledge that you have from previous assay development activities and the knowledge we've gained from studying viruses over the many decades, but usually you're looking for sequences and regions that are unique to the virus that you're targeting, so you don't have specificity issues, and that are highly conserved based on the function. So this was a rather nerve-wracking experience with SARS because we had to move so quickly with so little information, but worked out okay.
Thank you, Dr. Cloherty. Our next question here is, with increased pharma interest in HBV, do you see the relationship between HBV DNA and RNA changing? Oh gosh, my screen jumped. Okay, changing as new therapies targeting different HBV pathways become available? And would these studies need to be repeated with different drug targets?
Well, I think the studies will need to be drug target specific and actually drug specific. I think it's interesting to see. We'll have to wait and see. Basically, we're doing a lot of work looking at existing nucleoside analog therapy and new drugs that are coming to the market, and as they work in different ways, I think the relationship between HBV, RNA, and DNA, and in fact other markers, serological markers like surface antigen, e antigen, correlated antigen, are all going to need to be assessed, and it'll have to be done in a very specific way for the drugs as they have different mechanisms of action.
Thank you, Dr. Cloherty. Our next question here is your focus on clinical isolates, or will you also conduct surveillance in animals and the environment?
So our initial focus is on clinical isolates, looking in human populations. There's a lot of work adjacent to our program that's looking at animal populations where we'll be collaborating and having a line of sight. But I think, there's a lot of viruses that are in animals that may pose a threat, but there's no guarantee. It's a very difficult thing to tease apart which ones will make the jump across. So we're trying to really stay focused on what is significant clinically to try and catch them early in the human population and understand what's going on there closer to the threat.
Thank you, Dr. Cloherty. Our next question here is, why are RNA viruses more dangerous than DNA viruses?
Well, I think RNA viruses, they tend to cause more disease. It's not inherent in RNA versus DNA. I think basically we have to look at the viruses and the clinical consequences that they cause. We will be, looking at both RNA and DNA viruses in our program, as well as other pathogens.
Okay, thank you, Dr. Cloherty. Our next question, do you or your controllers have additional follow-up studies planned for the elite controllers study?
So we are looking at ways to study this topic more closely. Will require follow-up and longitudinal sampling from these patients, look at their immune system. These are things that are in discussion and see how we can go about making that happen.
And our next question, I applaud you for the initiation of the Pandemic Defense Coalition. Is there a certain profile of institutions you are looking for, and how does one initiate this type of collaboration?
So I think we outlined in the presentation there, kind of some of the characteristics. We need people who have access to clinical samples and interesting patient populations. Geography is important, expertise. We're looking for expert partners. And I think the way of connecting can be through local Abbott representatives or, directly to myself or the other members in R&D. But yeah, I think you can contact your local Abbott rep or scientific affairs or anybody to get in touch with us.
And our last question here for you today, your surveillance program uses NGS for SARS variant monitoring. What are your thoughts around different variant detection assays? And is Abbott developing a variant or a sequencing assay?
So I think, sequencing is critical in looking for new variants and because when if you've got a test that's specific for certain mutations that have been strain defining variants or mutations, then you will miss anything else that's out there. So sequencing remains kind of the backbone for looking at emerging new variants. I think you can develop assays to pick up specific mutations. That's only really clinically relevant if you're going to do something with that information in terms of treat people differently, quarantine, behave differently. So right now, those are kind of epidemiological tools. I think there's a few of them that are coming out, and so we're looking at that. In terms of whether or not we would build one, it really depends on the clinical utility, in terms of moving it forward in a regulatory process as opposed to a research tool.
Thank you, Dr. Cloherty. Do you have any final comments for our audience?
No, I'm very grateful for people who took the time to spend this time with us and participate in this call, for the interesting questions that they posed. And we're very open to collaboration. If anybody wants to reach out, as I mentioned, through scientific affairs, local reps, or directly to us, we look forward to hearing from you.
Thank you again, Dr. Cloherty, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Global Scientific Affairs Molecular Diagnostics at Abbott, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.